All right. So last class, while I was away, you all started the protein trafficking lecture and got a chance to go over and discuss what the regulation and trafficking mechanisms look like for co-translational import. We're going to finish up. We're going to finish up this lecture. Bless you, bless you. And we're going to move on after we finish this lecture up to go and begin the unit, or not unit, the last series of lectures on metabolism. And so we're going to finish up this lecture discussing the post-translational import that's associated with nuclear localization sequences in the mitochondrial target sequences. And so typically what ends up happening here is that the proteins that are being trafficked up to the nucleus or into the mitochondria, what ends up happening here is that these are going to be proteins that are uniquely tasked with functions that situate them within the nucleus or situate them within the mitochondria. But what that means is that they need to be moving up a concentration gradient. And so they're going to need assistance or help with moving up that concentration gradient. And they're going to be leveraging another protein's concentration gradient that's going to serve as a carrier to help them get there. So first, we're going to be talking about what it looks like to import some of these proteins that have the nuclear localization signal to get them into the nucleus. And so these nuclear proteins possess this sequence, the nuclear localization signal, that indicates that they are tasked with and destined to be in the nucleus, to be a nuclear protein. These proteins end up being made by free ribosomes that are not bound by the ER. And what ends up happening here is they're going to be using another protein's concentration gradient to help get them into the nucleus after translation. And that protein is called important. Important, what it does is it actually binds to the nuclear localization or the NLS sequence on this protein that was translated. And what ends up happening is that the nuclear pore complex here recognizes the important protein complex. And once it recognizes it, it enables it to go to be transported into the nucleus. Once that happens, important has an affinity for this other protein called RAN, and so it'll disassociate from our cargo protein here, and important will go over and bind to RAN, and it'll be carried by RAN out of the nucleus back into the cytosol, where it's this RAN GTP. The presence of GTP is important because once it's transported back out of a nuclear pore protein into the cytosol, once GTP is hydrolyzed to GDP, it will release important. So the key thing to note here is that important will bind to our protein that's translated, that has this NLS sequence. It'll carry it, and so importance concentration gradient carries it into the nucleus. So our protein here there's two concentration gradients that work here. There's a concentration gradient for the protein with the NLS, and it needs help moving up its concentration gradient into the nucleus. It's leveraging the concentration gradient of important. Important will migrate into the nucleus, and so they'll go in together, and then important will be shuttled back up its concentration gradient out of the cytoplasm by associating with RAN that has a GTP on it. Once that GTP is hydrolyzed, important will be released from RAN. So this is how post-translational import occurs into the nucleus. The export process is pretty similar. For the key thing of why we'd want to export things out of the nucleus, one thing that you guys, I think, discussed about last class was how proteins that are under 40 kilodaltons can freely diffuse into the nucleus. And so we typically only want nuclear proteins to be in the nucleus, and so we need a mechanism to transport them out of the nucleus, these free proteins that can diffuse in. And so there's going to be proteins that have this nuclear export signal that's recognized, and it's going to be recognized by exportin. And so to help you remember important versus exportin, it's kind of in the name. Important helps import things in the nucleus. Export helps to export things out of the nucleus. And so exportin will bind to this nuclear export signal to the NES, and that's going to happen up in the nucleus. And so exportin is going to be also bound to RAN here that has the GTP associated with it. So the protein with the NES bound to exportin, bound to RAN, this whole complex will together ride down the concentration gradient. And once GTP is hydrolyzed, it'll release exportin and it'll release this protein that's now properly within the cytosol, not in the nucleus anymore. And exportin will now ride its concentration gradient back into the nucleus. Are there any questions on these two things here? Yes. So proteins under 40 kilodaltons, fairly small proteins, are able to freely diffuse across the nuclear membrane into the nucleus. And so this is just a mechanism to make sure that those small proteins that are under 40 kilodaltons can be properly escorted out of the nucleus when they shouldn't be there. Yeah. That's just the RAN. You can associate the hydrolysis of GTP. It's only happening within the cytoplasm. So like in that context, GTP will be hydrolyzed, and that'll induce a conformational change that will then prompt exportin or importin to be released. All right. So this is a better representation of what the nuclear pore complex looks like. It's a really large structure, and as with a lot of other things we've talked about throughout the course of the semester, characterizing and identifying the components and features of the nuclear pore complex, I believe won the Nobel Prize a short while back. But it's this large 50 megadalton protein assembly that has 30 distinct NUPs or nuclear porins. And so you can appreciate that there's a series of features of small inner ring proteins that are stacked around each other, all characterized by these different kinds of NUPs. And there are over 450 individual proteins within a single nuclear pore complex here. So up to this point, we've talked about how there are some proteins that are tagged or modified so that they are bound and restricted to the cell membrane, whether on the outer leaflet or the inner leaflet. We've talked about how with G proteins, the alpha and beta and gamma subunits are really bound to the inner part of the cell membrane. They'll move across the cell membrane to activate and bind to other molecules, but the way in which they're bound to the cell membrane is going to be a post-translational modification that we talk about here. And so the way in which proteins are targeted to be membrane bound is by a modification called prenylation. And so what ends up happening is that on the C terminus, there's going to be a motif. Again, motif is like a specific amino acid sequence here. It's cytosine, alanine, alanine, or cysteine, not cytosine, cysteine, alanine, alanine. And then X is any amino acid after that. And they call it the CAX box. So the CAX box is a motif that is a target for prenylation. These prenyl groups are these long hydrophobic hydrocarbon tails that are able to be embedded within the cell membrane and enable these proteins to be modified to be tethered to the inner part of the cell membrane. And so any prenylation is going to tether it to specifically to the inner leaflet. We're going to talk about an outer leaflet change, but prenylation is for the inner leaflet on the C terminus. So when we have two prenyl groups, each of these is a prenyl group. So when we have two prenyl groups, we call that a journalation. When there are three prenyl groups, we call that a farnesylation. And when there are four prenyl groups here, we call that a journalation. And one modification that tethers a protein to the outer leaflet is when we have a glycophosphatidylinositol anchor or a GPI anchor. It copies some of the same phospholipids that are on the outer leaflet there, but it's a way in which some proteins, bless you. Seems like people are getting allergies and a lot of sneezing today. A GPI anchor is how a protein is tethered to the outer leaflet here. And so like for a protein that I've been studying for the last 5 years, that has a GPI anchor, and it's a way in which some people can study the way in which it can sometimes regulate or imbue a special quality to some of these proteins. And the way in which they can be cut or untethered is via phospholipase. Another inner leaflet modification is myristylation, but that will end up happening on the N-terminus. And as I'm sure everyone remembers here, on the N-terminus, where the basically the front end of an amino acid sequence or a peptide, the first amino acid is going to be a methionine. And so the target for a myristylation is a methionine glycine on the N-terminus, and that'll tether a protein to the inner leaflet. So last class, I think you all spent some time talking about the RAS, RAF, MAPK signaling pathway as it relates, and we're going to talk about that as it relates to Noonan syndrome today. But RAS is a G protein that's tethered to the inner leaflet, and it's farnesylated. So it means that it's going to be tethered with three prenyl groups to the inner leaflet of the membrane. And so that's how it's restricted there. And so this prenylation, what it actually looks like here is that there's going to be on the cysteine. So again, we said that there's a CAX box that's a target for this prenylation modification. And so the cysteine here creates a disulfide bond. And we talked about these before. It's this really strong covalent bond that's created between two free thiols. And so this disulfide bond is a really strong covalent modification that keeps that protein there embedded within the inner leaflet there. So with myristylation, what ends up happening here is that there's going to be an irreversible addition of a myristoyl group to the N-terminus. And we talked about how that's going to be happening specifically. It's targeting methionine glycine residues on the N-terminus, the front end of a protein. And that's catalyzed by specifically a myristoyl transferase enzyme here. And so how to bring this all back to Noonan syndrome, what ends up happening is that when we look at the RAS, RAF, MAP kinase signaling pathway, we have RAS that's going to be bound up here as a G protein to the inner leaflet of the membrane. And it's going to, once it has a GTP molecule, it's going to be able to interact with RAF here. But again, RAF is going to be a cytosolic free protein, and RAS is a G protein stuck to the inner leaflet of the membrane. And so for that to freely diffuse and for the two of them to interact with each other, it needs some help, for lack of a better phrase, for this free diffusible protein, the cytosol, to interact with a membrane-bound protein. So there's a scaffold protein called SHOC2. Scaffolding proteins typically are proteins that bring two other proteins together, for lack of a better phrase. And what's going to end up happening is that SHOC2 will help escort RAF up to RAS there and enable their interaction for this signaling cascade to occur. Now, what ends up happening in Noonan syndrome is that this scaffolding protein, SHOC2, again, scaffolding protein, SHOC2, just means that it enables the interaction of two other proteins. What ends up happening is that there is a mutation in Noonan syndrome with SHOC2. So we have here, it says serine 2. That means the second amino acid is a serine. So the mutation goes from a serine to a glycine. So if the second amino acid is a glycine, do we know what the first amino acid is? It's not a trick question. I've already given the answer. It's the same for every protein. Methionine. So this mutation, I know you all were thinking in your head, so I just said it out loud for you. So the fact that it goes from serine to glycine now makes it vulnerable to a myristylation modification, and that's exactly what happens here. And so this myristylation that happens as a result of this mutation will now tether SHOC2 to the inner leaflet of the membrane. And so now this will create a more chronic activation of this RAS-RAF-MAPK signaling cascade. And so, and the MAP kinase signaling pathway is tied to a lot of important biological pathways and processes. And so the chronic activation of it is going to be tied to a lot of different outcomes here. And so that's the explanation of what happens to Noonan syndrome. There's this mutation in SHOC2 from a serine to glycine that makes it susceptible to a myristoylation event, and that myristoylation will tether it to the inner leaflet of the membrane, driving chronic activation of this signaling pathway. If you're interested in protein trafficking, Jeff Sosserman he combines computational modeling with actual signaling events as it relates to protein trafficking, and David Castle is another researcher in the Department of Cell Biology. As I'm getting ready to pivot to the next lecture, are there any questions here about what we've discussed? Yes.
Can you describe the difference between prenylation and the myristoylation?
Yeah, so prenylation, prenylation recognizes specific motifs, specific amino acids, the CAX box, CAAX, cysteine, alanine, alanine, and then X is any amino acid. That's on the C terminus, on the end. Myristoylation recognizes methionine, glycine on the N terminus. So one thing that's common about them is that they'll tether a protein to the inner leaflet of the membrane. So they both do that. How they're different is prenylation recognizes a motif on the C terminus. Myristoylation recognizes a motif on the N terminus. And they both will add like a hydrophobic chain to each end, to the end that they target, and tether it to the membrane. Great question. I'll go here and then I'll go there. Go ahead.
Why do they only target either way?
Yeah, so that's a great question. I'm not 100% sure, but what I suspect is that we know that in some cases, specific phospholipids are restricted to the inner leaflet versus outer leaflet. And so I suspect that the way in which these specific hydrophobic tails are recognized, or the way in which they're embedded, the mechanism by which that happens, I think that mechanism restricts them specifically for these, for a GPI anchor restricts it to the outer leaflet, just because I think the features of a GPI tail matches some of those outer leaflet restricted phospholipids, and the same for the prenylation or myristoylation. Yeah, that's a great question. I think that's the case. Again, this is a class of half-truths, and so the second half, I think that's the answer. Yeah, go ahead. Both proteins. RAS is the G protein that's tethered. So this is a good question. RAS is the G protein, and because it's a G protein, it has this modification that tethers it. It's the farnesylation that tethers it to the inner leaflet. And then RAF is just a soluble protein in the cytoplasm that interacts with RAS, and that has SHOC2 help that interaction. But you can think of, but RAS is upstream. It goes RAS that interacts with RAF, and then RAF interacts with subsequent kinases below that drive signaling and transcription. Last one, go ahead.
So why does the SHOC2 protein being on the membrane cause that?
Yeah, great question. I should have jumped into more detail on that. Great question. So the SHOC2 as a scaffolding protein helps enable the interaction between RAS and RAF. It makes it more easy for those to interact with each other. And so but what that requires is both SHOC2 to be close to RAS and for it to interact with RAF as well. So by it being now bound to the cell membrane, like in the same domain where RAS is, it makes it a lot easier for it to interact and activate RAS and RAF. Because if you think about it, if we have two proteins that are kind of like freely diffusing that need to like meet with each other and then like meet with RAS on the membrane, it makes it a lot easier if only now like one of them is freely diffusible. If we have like three actors in this interaction, if two of them are now bound up here in like the same domain, it makes it a lot easier for this interaction to be occurring and for it to occur now chronically as opposed to being susceptible to some more other regulatory mechanisms. Does that help clarify? That was a really good question. I should have explained that better. All right, so now we're going to pivot and we're going to finish out the semester focusing on metabolism. And it's actually kind of weird for me to say that out loud, just because in some ways it feels like the semester still started. And but yeah, so we're going to finish out the semester with these last 2.5 lectures focusing on metabolism, focusing on different features of metabolism. And we're going to be talking about them from the context of obesity, diets, and alcohol. I'm going to first take a step back and say, I've tried to spend a lot of time updating some of the different slides because I think I've said to you all, I took this class now, weird to think this out loud, 14 years ago. There are some things in biology haven't changed. And because of that, some of these slides haven't changed. In addition to that, some of the disease wrappers are the same, some of them are different. This is one I have not yet taken the time to update. And so there are going to be points where I jump in and perhaps try and make addendums to perhaps update things that I think are a little out of date. So not because of biology, but rather how we think about these things, particularly as it relates to things like obesity and diet. So to give some context, obesity is simply defined as excess body weight. That's become a really important public health issue. It's been associated with a lot of different outcomes. It's been strongly associated with type 2 diabetes, with hypertension, which is high blood pressure, with stroke, with hyperlipidemia, that's just high lipid content within bloodstream, as well as some cancers. An obese patient historically has been identified as someone who's having 20% more or 20% or more weight than their ideal weight. Ideal weight is defined by BMI. So what I'm going to do is I'm going to hit pause right here and say BMI is not really the best indicator. Historically was used as a way to identify a concept like ideal weight. I am going to say I think ideal weight's not helpful, nor BMI is a really helpful indicator of what someone's weight should be relative to their height. A lot of studies have identified and shown that, and I think a lot of us can appreciate that there are some professional athletes that have like 5% body weight, but are considered by BMI either overweight or some of them obese, despite having really healthy lifestyles and really healthy habits. And so the concept of ideal weight and BMI are not very helpful as it relates to thinking about as it relates to thinking about health and weight. But BMI as a tool is typically identified by looking at a ratio of weight to height squared. And so, yeah, I've already said this, but it doesn't account for lean body mass. It doesn't account for people who have higher muscle mass. And so again, it's not also, I think the defining of what is ideal weight or what is considered normal isn't a really helpful paradigm, especially as it relates to how we consider helping people live healthier lifestyles. I'm going to get off my soapbox now. there was a study back in Scotland a while back that helped identify some of the positive outcomes that are associated with modest weight loss. Some of those outcomes showed pretty dramatic changes, including 25% reduction in premature death, drops in associations with diabetes-related complications, reducing risk of dying from cancer, as well as changes in both blood pressure, the lipid profile, as well as responsiveness to glucose as it relates to diabetes. And so this study is great and shows that proper weight control can be really helpful, but this does by no means substantiate or promote that BMI is a helpful paradigm to use when considering healthy lifestyles and when we think about weight loss. So the key things that we're going to be talking about throughout this metabolism unit are going to be some of the key steps and features of metabolism and how ATP is produced, how fat can be synthesized and both used as an energy substrate, some of the key major diet paradigms that historically have been adopted and how they work and why they work, the metabolism of alcohol, as well as the origins of some free radicals are important for metabolism. So we've talked a lot about ATP throughout the course of this semester. It's been a key energy resource for a lot of different reactions that we've talked about. And so the whole point, for lack of a better phrase, if I wanted to create a one sentence that identifies like the theme of these next 2.5 lectures, the whole point of our metabolism lectures is to try and discuss how ATP is produced and how it can be generated as a resource for the bioenergetics for everything we've talked about up to this point. And so ATP is consists of this these three phosphates in the phosphate group. There's this base sugar here, adenosine, there's a base sugar, there's ribose sugar here, and there's this base group adenine that all together makes adenosine. And we've talked about the process of ATP hydrolysis, whereby it loses a phosphate group that then can be used to activate certain pathways, or can be used via kinases to activate things. And some of those processes can either be kinases, pumps. And so what ends up happening is that ATP, we've talked about it so much, it's important to note that there's high concentrations of ATP throughout our cells that enable us to use them for a variety of, as a resource for energy for a variety of processes here. So we're going to be talking about how ATP is synthesized in 2 different contexts. We're going to be talking about them in the context of in an anaerobic context where there's an absence of oxygen, or an aerobic context where there is oxygen. And we're going to be talking about that briefly, about how that looks like in us, in mammalian cells, versus in some microorganisms like yeast. And so most of the anaerobic processes we typically refer to as like fermentation. We have lactate fermentation, or ethanol or alcohol fermentation, and those are going to be occurring in anaerobic contexts. And most of the times when ATP is going to be produced in mammalian cells is going to be leveraged via aerobic processes, because we're oxygen breathing mammals. And so what's important to know is that also there are certain tissues that operate in both contexts, in both aerobic and anaerobic contexts. As you all can probably appreciate, skeletal muscle can work in an anaerobic context where there's really high intensity that's in locally that has an oxygen deprivation that will then use lactate as an energy substrate to produce ATP. And also during intense cardiovascular workouts or cardiovascular events, there's local anaerobic events within the heart as well. So what we're going to do is we're going to try and we're going to try and focus on some of the key features that set everything up for these processes first. And we're also going to talk a little bit about define a few key things that I'm sure that all of you guys have already discussed, whether it is in chemistry, whether a few of the brave 30 years have already taken biochem. But to create a common foundation, I'm going to take a moment to try and describe oxidation and reduction here. And so the metabolic substrates that we're going to be talking about throughout the course of these next couple lectures are going to be highly oxidizable, meaning that they're going to be there's a word I'm trying to say here and I'm forgetting it. They're going to be able to be oxidized, which means it's a reaction whereby they lose an electron. And so what that's going to look like here is that, as an example, we have ethanol that undergoes oxidation, that becomes an acid aldehyde, that then will lose 2 electrons here. The reverse process here, whereby acetaldehyde will gain 2 electrons to produce ethanol, that would be a reduction event where it's gaining electrons. What's also happening, you might note that there are 2 hydrogen ions that were produced or lost as a result of this oxidation event. So while there are also the loss of electrons, there's going to be events that are losing hydrogen ions as well. This loss of hydrogen ions, we typically call dehydrogenation. And so, as you can imagine, the enzymes that mediate this dehydrogenation are called dehydrogenases. And so you're going to be hearing dehydrogenase throughout the course of these next couple lectures for some reactions. And the key thing to note here is that a dehydrogenase is an enzyme that mediates the loss of a hydrogen ion. In this event, when ethanol is being metabolized, this is mediated by an enzyme called alcohol dehydrogenase. So when we have the loss of these electrons, it's really key. These electrons become really key for the final events of particularly aerobic metabolism. There's going to be, their final destination will be eventually to an oxygen serving as an electron receptor at the end of metabolism. But in the meantime, in the interim period, there needs to be something that will carry those electrons to help them get to their final destination during metabolism. And so there are going to be these intermediate electron acceptors that will then also be donating and passing on those electrons. These electrons kind of almost serve as a bit of a potential energy for future energy, for lack of a better phrase, for future ATP. And so these intermediate carriers of these electrons are coenzymes. And what ends up happening is that these coenzymes help mediate the function of enzymes during metabolism. And the most common coenzyme that serves as an intermediate carrying these electrons is NAD, nicotinamide adenine dinucleotide. We're just going to call it NAD or NAD plus from here. We'll also be calling it, there's also going to be a form of it that we'll refer to as a reducing agent, and we'll discuss that on the next slide here. So when NAD accepts both a hydrogen ion as well as 2 electrons, it'll become NADH. That's the reducing equivalent. It's reduced. It's gained electrons here, and so NADH is the reducing equivalent here. And so what ends up happening is that there's going to be electrons that are stored in NADH as a potential energy, as a future resource to help reduce oxygen for the future production of ATP in aerobic metabolism. And so for the structure of NAD, we have our 2 phosphate groups here. We have adenosine. A lot of this is going to look familiar. We have a ribose sugar here, but then there's going to be this nicotinamide here that's currently in its oxidized form. And what's going to end up happening here is that when it's reduced and gets 2 electrons in the hydrogen ion, this double bond is going to be reconfigured to enable 2 hydrogens to be added on, to be present on this ring here. So nicotinamide, so this depicts it in its both oxidized and reduced form. Nicotinamide becomes a really critical metabolite. And what's important to note is that for a key feature or a key quality of our diet is going to be vitamin B3, which serves as a precursor for the production of nicotinamide. So before, I'm going to stop for a couple of questions here. Are there any questions on what we've discussed so far? Yes.
Can you repeat that last part about going to
Yeah, so there's going to be a final destination for the electrons that are going to be lost as a series of the oxidation reactions that occur throughout metabolism. And those electrons are going to be used at the end of metabolism. But we need something to kind of carry those electrons before they get to their final destination. And that's what NAD does. It will take on hydrogen ion and these two electrons. A single NAD will take on one hydrogen and two electrons and become NADH. That's its reducing equivalent. And so it's accepted electrons here. And then as we move down metabolism, this is kind of skipping ahead. As we move down during metabolism, it'll then donate those electrons and it'll lose them. And its final destination is an oxygen at the very end of metabolism. But there's going to be a number of NAD molecules or NADH molecules that are going to be important across different steps of metabolism here. Yep. Are there any other questions? Yes.
What does it mean reducing equivalents? Is that referring to NADH or the reaction?
It's referring to NADH. So that's the form that has gained an electron. A trick that I learned to help remember oxidation versus reduction. You guys may have learned this. I might be dating myself here, but there was a little term oil rig that I learned, I think it was even back like in high school. Oil, oxidation, L loses, reduction, R, G gains. So like oxidation loses an electron, oil, rig, reduction gains an electron. You guys may be totally tired by the end of the semester right now, so but yeah, so that's a small shorthand that I learned that helps me remember oxidation versus reduction, and what's happening with the electrons in each of those reactions. Last question, and we're going to press on.
Vitamin D is before producing NAD?
It is an important precursor that helps with the production of, I believe it helps with the production of nicotinamide, that part of NADH here. All right, so we're going to jump into glycolysis and fermentation. So glycolysis, the entire goal of glycolysis is to take a glucose molecule and use it as a substrate to produce ATP, to yield energy. And so the opposite of glycolysis is actually a pathway called gluconeogenesis. Glucose, new, and genesis, generation or production of it. And so gluconeogenesis is an opposite pathway where it takes other substrates and tries to produce new glucose. And so glycolysis typically is not an oxygen-dependent part of our metabolism, and so it is an anaerobic portion of our metabolic pathway. And what ends up happening is that what's produced at the end, it's going to be a molecule called pyruvate. What's produced at the end will then be broken down further, depending whether we're in an oxidative or non-oxidative environment, whether we're in the presence or absence of oxygen here. We've already referred to this really briefly, but another key step in metabolism is fermentation. We have lactate and alcohol fermentation. Those are also going to be anaerobic pathways that involve the breakdown of a nutrient molecule of pyruvate without any oxidation events happening. And so what ends up happening, what's key here is that what's key about the fermentation is that if there's output of glycolysis, if pyruvate builds up enough without the presence of oxygen, there needs to be a way to handle to try and handle that substrate. And so that's what fermentation enables here. So we're gonna try and go through the different steps of glycolysis, and I'm gonna expect you guys to memorize every. I'm just kidding. I'm sorry. I don't want to do that to you right now. I couldn't even say it straight face. So there are a lot of steps in glycolysis, a lot of intermediate metabolites, a lot of enzymes that produce those metabolites. What I'm going to ask of you is I'm going to try and bin some of these steps together, and I'm going to ask you to remember, for lack of a better phrase, these bins, these like like overarching parts that will kind of lump together some of these steps. And I'm going to ask you to also note at the very end, we'll talk about some like regulatory parts or some like key features here. So like, I'm trying to avoid saying like, this will be on the quiz or like this will be on the exam. There's going to be some like key qualities or key features or some key things that I'm going to try and identify and highlight. Whatever I'm highlighting, I want you to take note of those things. I'm not going to ask you to memorize every single enzyme. So the first steps of the whole process of glycolysis is going to be breaking down glucose to produce energy. And so glucose can either be represented in its 6-carbon in its ring structure here. Typically, there's also another way it can be denoted in this linear orientation as well. Typically, the linear orientation helps us to appreciate what the changes are, and sometimes as well some of the symmetry that's going to be key to note here. And so starting from here, we're going to be focusing on glucose and the intermediates and their linear orientation, but they can be represented both ways. And so what's going to end up happening is the 3 key steps that we'll be talking about are the preparation and cleavage of glucose, a key oxidation event, and the eventual formation of pyruvate. And so when you guys take, for those of you who end up taking biochem, this would be just like this is pulled out straight of a biochem textbook. You'll likely be expected to memorize every feature, and I think also draw out a lot of the parts of glycolysis here. So that's just to both mentally and emotionally prepare you for that. So the first step is preparation and cleavage here. So what we're going to be doing is we're going to be starting with a glucose molecule here that's depicted. And so what ends up happening, this first step, is that there's going to be a phosphorylation event where we lose an ATP molecule here. So that's what's going to be key to note here is that there's going to be ATP put into the system that will then help us produce ATP on the back end. But we're going to be starting here first with expending an ATP molecule, and we're going to be taking the lost phosphate group there and adding a phosphate group here to glucose to produce glucose 6-phosphate. There's going to be an isomerization. There's going to be a changing, not chemically, but changing the structure of glucose 6-phosphate where it becomes fructose 6-phosphate here. What I should say here, sorry, is that this first enzyme that mediates the addition of this phosphate group is called hexokinase. And the isomerization event is mediated by an enzyme called isomerase here. Now, what ends up happening is that there's going to be a second ATP molecule that's put into the system, and that phosphate group is going to be used to add a phosphate here via an enzyme called phosphofructokinase. So now, if you can appreciate, there's a bit of a symmetry here of this fructose 1,6 biphosphate here. And so this symmetry enables fructose 1,6 phosphate to be cleaved via an enzyme called aldolase here. And so now we have two molecules here, two molecules of glyceraldehyde 3-phosphate or G3P. So for the preparation and cleavage, it was phosphorylated, isomerized, phosphorylated again, and cleaved. So we put into the system here 1 glucose and 2 ATP to produce or yield 2 G3Ps, and we have 2 ADPs on the other end. And so the final, what's key to note here is that we're putting one 6-carbon one single 6-carbon ring into the system, and we're going to be producing 2 3-carbon chains here. And so we already have our 2 3-carbon chains here in the form of G3P. So the next step is going to be, I'm jumping ahead of my own notes, is going to be a series of steps that involve oxidation here. And so at this point, what I want to try and focus, what I want to try and definitely highlight is we talked about, we produced 2 G3Ps on the previous page, 2 glyceraldehyde 3-phosphates. We're going to talk about them now one at a time. So everything on this page is happening twice because we produced 2 G3Ps. But now what we're going to do is we're going to talk about 1 G3P at a time. But again, if you're taking note, if you're taking inventory of the energetics here, everything you see on this slide is happening twice. So what ends up happening here is that there's going to be an oxidation event of G3P. And so we have our first NADH. We have our first production of a reducing equivalent, whereby this phosphate group, or sorry, not phosphate group, forgive me. There's going to be 2 electrons and a hydrogen that's going to be donated to the NAD to produce the reducing equivalent. And this oxidation event is going to be happening via an enzyme called glyceraldehyde 3-phosphate dehydrogenase, or GAPDH. And so what ends up happening next is we have we produce the 1,3-biphosphoglycerate. And what ends up happening here is we have our first production of ATP here. And again, all of this is happening twice. So we've produced 2 ATP here. And this is going to be a key thing here is that this is going to be an example of where the phosphate group from our previous metabolite will be used to phosphorylate an ADP. And so this is an example of substrate level phosphorylation, whereby we're able to produce this ATP. And once this happens, the subsequent metabolite is 3-phosphoglycerate. And so to take inventory of the energetics here, for each G3P, we're going to produce 1 3-phosphoglycerate, 1 reducing equivalent, NADH, and 1 ATP. So this is happening twice, again, because we produced 2, we've done this twice based on what was produced on the previous slide. And so this, to try and focus on the take-home message here, because I think there's, I see a lot of eyes glossing over here. The key take-home here is that there's an oxidation event that occurs where we're able to produce both an NADH and a subsequent substrate level phosphorylation event that enables the production of our first 2 ATPs. Again, this is happening twice because we've produced 2 G3Ps based on the previous page, based on the preparation and cleavage. So the final slide on glycolysis, just to help you guys, help you see the light at the end of the tunnel. So we have, for each 3-phosphoglycerate, what's going to end up happening here, and we're going to be moving on the bottom from right to left here. There's going to be a mutase event, forgive me, whereby there's going to be a rearrangement of 3-phosphoglycerate to 2-phosphoglycerate. And then there's going to be an enzyme called enolase here that ends up with the production of a phosphoenol pyruvate here. And then again, there's going to be another substrate level phosphorylation event whereby the phosphate group from phosphoenol pyruvate will be used to phosphorylate ADP for it to become ATP, and then that leaves us with pyruvate. So for each, so to like take a step back, take home message on this slide is that there's going to be from 3-phosphoglycerate, we're going to get 1 pyruvate and 1 ATP. There's going to be, and again, this is happening twice for each molecule of glucose. So now, if we take a step back and look at the inventory from glycolysis, for each molecule of glucose that we have, we're going to yield 2 pyruvates. We're going to yield 2 NADH, which is the reducing equivalent here, and 2 ATPs. And so if you remember, we had to put we actually are producing 4 ATPs, but we put 2 ATPs into the system, which is why and again, the key thing is net output from the title of the slide. The net output is 2 here. And so what ends up happening from here after glycolysis is going to depend on whether or not oxygen is present here. That's going to inform what happens with pyruvate. And so in the presence of oxygen and during aerobic respiration, there's going to be an enzyme called pyruvate dehydrogenase whereby there's going to be it's a dehydrogenase, so there's going to be an oxidation event. A key thing to help note here to try and help decipher things. If you see, we talked about this earlier on in our first oxidation slide. If you see a dehydrogenase or if you see a dehydrogenation event, that indicates an oxidation event here. So pyruvate dehydrogenase helps mediate the oxidation of pyruvate, and it also produces a carbon dioxide molecule via coenzyme A. And the production here is acetyl-CoA. So put a pin on this. Like this is going to be like a key thing for us to consider moving forward. For aerobic respiration, we go from pyruvate to acetyl-CoA. We're going to talk about that next class. In the absence of oxygen, we get fermentation here. For mammalian cells, what ends up happening is from pyruvate, there's an enzyme called lactate dehydrogenase here that produces lactate or lactic acid. And so that's going to be used as a substrate for anaerobic respiration. Lactate is, and some of you might have heard lactate buildup in your muscles. That's typically happening during a high intensity event where there's local deprivation of oxygen. Fermentation in yeast or some other microorganisms, instead of lactate fermentation, you'll actually have alcohol fermentation. There's going to be an enzyme here, pyruvate decarboxylase, where there's the production of a carbon dioxide molecule. So this is why beer and sparkling wine, champagne are fizzy, because there's a carbon dioxide molecule that's released during the metabolism of each pyruvate molecule. And so particularly this will be happening in yeast. And so there's an intermediate called acetaldehyde here. There's acetaldehyde dehydrogenase that'll then produce ethanol. A key thing to note here is that the concentration of each of these metabolites will determine which way the reactions are going. And so if there's a buildup of pyruvate, it'll move towards ethanol metabolism. And if there's a buildup of ethanol here, for example, if you consume ethanol, which you're not going to do when you're working on your first draft of your research statement that's due tonight. With a buildup of ethanol, it'll then be metabolized into acetaldehyde and then metabolized into pyruvate. And if you're thinking to yourself that ethanol is being used in the production of an energy metabolite, hold that thought, because we're going to talk about in a couple lectures how ethanol can be used to excess alcohol can be used to generate excess fat, which is great to hear, I assume. I already just said all this. Okay, so when we have the buildup of lactate, either in our skeletal muscle and cardiac muscle, what's going to end up happening is that there are transporters on our cells that'll then shuttle lactate out. It'll then be transported via your bloodstream out to your liver to be used as a substrate to produce glucose here. So this is an example of gluconeogenesis. It's taking glucose and it's using it to produce. It's taking lactate and using it to produce new glucose molecules here. And so this is how, during recovery, when there's lactate buildup in your muscles, it can be used to produce glucose as a subsequent energy substrate for downstream glycolysis. So before I talk about some of the last key take-home points of regulatory features of glycolysis, I'm going to step back and give space for a couple questions here. Yes.
Can you just give a summary of
Yeah. So the take home here for the Corey cycle is that lactate buildup that occurs in your skeletal muscle, particularly, this might have been a helpful context. The acidification of your skeletal muscle, it doesn't want that lactic acid to be built up, so it needs to be shuttled out and used elsewhere. And so it'll be transported out of these skeletal muscle cells, which will then go to your liver, and your liver will use lactate and use via the process of gluconeogenesis, the generation of new glucose. That gluconeogenesis will use lactate to produce a glucose molecule. And now that glucose molecule will leave the liver and go to cells that require energy, require glucose to produce energy. So it's basically like from a 30,000 foot level, taking lactate to produce glucose. Yes. That's typically how I think of it. Again, like there's also local oxygen deprivation that can happen in cardiac muscle as well. But I feel like it's most commonly talked about in skeletal muscle. I'm trying to think if there's like also like an example of like the flight or fight response. No, that's not. No, I'm gonna step away from that. You can just think about this particularly as it relates to like high intensity events with skeletal muscle. All right. So we're going to talk about the last key regulatory features of glycolysis to bring us all home. So I briefly referenced to this on the fermentation slide, but the steps that involve glycolysis and gluconeogenesis, they involve a lot of the same steps. And what actually determines which direction these reactions are going are largely based on the concentrations of these metabolites. And so if there's sufficient concentration of, say, glucose, it'll move down this pathway that will pop up on the screen any second now. There's sufficient buildup of glucose, it'll move down this metabolism pathway that's glycolysis. Whereas if there's a buildup of certain metabolites that drive the reactions back upward, that'll promote gluconeogenesis. But what's key to note is that there are certain steps where once you cross that Rubicon, you can't go back upwards. And so there's going to be three key steps that are largely highlighted here by the enzymes to the left of the signaling path of this metabolism pathway. And so the first irreversible step is glucose phosphorylation by hexokinase. And so once there's enough buildup of glucose, hexokinase will phosphorylate it. It cannot simply, with enough buildup of glucose 6-phosphate, promote the reversal of that process. And another key thing to note is that a lot of these irreversible steps also particularly relate to phosphorylation events as well. And so the other irreversible step here is at the level of phosphofructokinase, whereby fructose 6-phosphate is phosphorylated to produce fructose 1,6 biphosphate. And so these are the first two irreversible steps whereby once you cross the Rubicon, you can't go backwards. Now, once we get down here below the production of fructose 1,6-phosphate, the changing of concentration can drive these reactions up, back and forth, depending on their concentration here. But the last and final irreversible step here is the substrate level phosphorylation of pyruvate, whereby we produce an ATP molecule to yield pyruvate here. Now, you can appreciate on the left that there are other mechanisms by which, via gluconeogenesis, we can go back up this pathway via gluconeogenesis, but it's leveraging other enzymes or other processes that can produce those other metabolites. So one example is to go back from pyruvate to phosphoenol pyruvate, we don't simply just go back up this pyruvate kinase pathway. Instead, there's a decarboxylase that turns pyruvate into oxaloacetate. We're going to talk about that later. So that name might sound familiar. And then from oxaloacetate via this other carboxykinase can produce phosphoenolpyruvate. So this is just an example of how there are mechanisms whereby gluconeogenesis enables us to go up that pathway, back up some of these irreversible steps, but it's not directly using these same enzymes. It's other processes that enable us to go back up. So in events where there is sorry, yes, go ahead.
So for glycolysis, there are the irreversible steps that you can change like from like between the steps, but you can't like between the steps again.
Yeah. Yes, that's actually you put it much better than I did. The irreversible steps are particularly as it relates to glycolysis. Gluconeogenesis leverages other enzymes to work back up the pathway, but we're not going to be using those same enzymes on the irreversible steps.
That's the one. That's the where they differ. Glycolysis is locked within the irreversible step.
Mhm.
It can only go forward.
Yeah.
With gluconeogenesis and go backwards and forwards.
I'll say yes, go backward and forward, but I would say it would switch. It would no longer be gluconeogenesis. Well, yes, I would say it can go backward and forward as like concentrations are changing during the process. But zooming out, it's the whole process of gluconeogenesis is definitely moving up. I would say where like the most changing is occurring as it relates to like up and down that you're referring to is largely in this middle portion here between fructose 1,6-biphosphate and phosphoenolpyruvate. That's mostly where this like swinging back and forth can occur depending on concentrations. That was not the most convincing K I've heard. That's if it doesn't make sense, that's okay. But was there a specific part that I did not do a good job to clarify?
I'll just make sure I highlight it.
Mm-hmm. Yeah. Are there any other questions? There's only a few minutes left, and the rest of it is a lot more straightforward, I guarantee you. All right, let's go ahead and wrap this up with the last key part. And so when we have excess glucose present, there needs to be a way in which that we can store it for future use. And so the way in which we do that is that excess glucose is stored in a polymerized form as glycogen, and it's particularly in the muscle and liver. And so what ends up happening is that once glucose is phosphorylated, it'll go from glucose 6-phosphate to be isomerized to glucose 1-phosphate. And then there's this uridine diphosphate, UDP, that will then be added on via an enzyme called UDP synthase. So the combination of UDP with a single glucose molecule here is the first building block of glycogen, whereby there's going to be the addition of subsequent glucose molecules to this building block here via glycogen synthase, where subsequent glucose molecules will be polymerized together via glycogen synthase to produce glycogen. And so glycogen is just the combination via these two steps to produce chains of glucose molecules that can be stored in liver and muscle as a future substrate for glycolysis. And so when the time comes where stored glycogen needs to be used as an energy source, what ends up happening here is that there's going to be a glycogen phosphorylase that's going to be then adding a phosphate group and cleaving a glucose molecule to produce glucose 1-phosphate. There's going to be an isomerization that changes it to glucose 6-phosphate, or not isomerization, a mutase, forgive me, a mutase that changes it from glucose 1-phosphate to 6-phosphate. And that then will feed into that second step of glycolysis that enables to use glycogen as an energy resource. The Last thing that I'm about 1 minute over, I'm gonna say this in about 30 seconds, is that there's feedback control on glycolysis here. And it matches up with the irreversible steps that we talked about a couple slides ago. So at the level of hexokinase here, hexokinase is, there's negative feedback once the concentration of glucose 6-phosphate begins to rise. So with enough, as this concentration begins to rise, it'll feed back and negatively regulate hexokinase to start turning it off. Additionally, once we start to produce enough ATP here, this ATP will come back and act on phosphofructokinase to start turning it off as a negative feedback mechanism. So these are two instances where the downstream products will act back up on an enzyme to start turning it off once there's enough downstream product produced. But also, there's one example of a feed forward or positive feedback loop, where as enough fructose 1,6-biphosphate is produced, it'll act down on pyruvate kinase to turn on that enzyme to promote its activity in subsequent production of pyruvate. We're going to stop there, and we'll pick up with the next parts of metabolism in the next class. And I'll be sticking up here if you have any questions.